Hello friends, welcome to my channel Plant Science for You. In this video series, we are discussing about different biometric techniques in plant breeding. And in this video, we are going to discuss about experimental designs. That means in plant breeding or in agronomy, we are using different field experiments. For that, we have to use different experimental designs like RBD, CRD, LSD, etc., which we will going to discuss in this video. So please subscribe my channel and like the video if you. So let's first understand what is experimental design, and before understanding about the experimental designs, we have to understand first what are the treatments. Treatments are those objects or those uh, character or those particular things for which we are going to see their effect on a particular crop. Okay, suppose we are checking what amount of irrigation will be perfect for the crop. We are testing different uh, amount of irrigation, or suppose different amount of nitrogen fertilizer, and checking their effect on the particular crop. Then this number of this number of criteria will be called as treatment. Okay, and as you know, what is an experiment? Experiment is basically a scientifically planned method. And what does design means? Design means designing or allocating or arrangements of different things. Like in our house, when we made our new house, we make arrangements of different things, like where there will be bedroom, where there will be a storeroom, where there will be our study room. We allocate them now. So the same is the case in case of experimental designs, where we allocate we actually what we do, we allocate different treatments to study or to draw a valid conclusion of a particular problem. Okay, or basically to study the effect of a particular problem or treatment. Then one thing comes in our mind, that is the what is the experimental unit. Experimental unit is basically a group of material to which a treatment is applied in a single trial. That means that are actually our crop, in which, or for example, we can say that the plot of land where the crop is grown, the group of cattle where the study is going on, or suppose a patient in hospital, the amount of dose of the medicine we are studying, its effect. So this will be called as our experimental unit. That means which we use as our testing material or substrate. We call it as an experimental unit. Now, if we see what are the three basic principles involved in studying and experimental designs, these are basically we use to reduce the experimental error and make our experiment more efficient. For that, we use three basic principles. What are those? Let's see one by one. First one is the replication, then there is a randomization, and the third one is a local control. What does actually replication means? You all know that replication means the repetition of treatments. That means one treatment is we are using more number of times. It will increase in the if we increase the replication number, that will increase the precision by reducing the error to a great extent. Okay. After that, what is the randomization? Randomization means random allocation of treatments. You all know that in the field there may not be the homogeneity in all the places. Somewhere there may be more fertile place, somewhere there is no less fertility. So what we can do, we can allocate our treatments in different location and random. Okay. In case of local control, what we do, we use something like greater homogeneity in experimental unit. That means if we use small plot size, in between that there will be less heterogeneity. We can say if there is a late heterogeneity is present, we can compare them with other treatments in very easy way, or we can say we can control, or we can reduce our experimental error. Suppose there is a field, and in that field, if there is a suppose this is a field, if in this field there is a heterogeneity, or you can say there is a fertility gradient is present. That means fertility is increasing from one end to the another. There is a gradient. We can use the term replication for reducing the heterogeneity. Okay, but if the fertility, suppose the fertility in the field present is in patches, that means here is a high fertile patches. So what we can do, we can just do the randomization. We can allocate the things or treatments in a random way so that they can be allocated in all the places. That means if sample number A is present, it will be present in high fertile region and also in low fertile region. Okay, and in case of local control, what we do, if this is our plot, we make them very small plots. We divide them into very small, and in this, suppose here is a two treatments, and in these small plots there will be very less heterogeneity we can expect. Okay, so what we do, we divide them into small plot size, and that is the way we can reduce the experimental error and increase our experimental efficiency. So let's start with our basic design, that is the completely randomized design, which we will use in case of pot culture or in the lab experiments only, which we call as the general term, that is a CRD. Okay, and it is important for lab experiments only. When we use this design, the conditions for use is the when there is a experimental material is limited in number and the field is homogeneous. Okay, what we do in the layout, we just divide our whole field directly into different small number of plots of equal size. Okay, and the number of plots will be equal to the product of the replications and the treatments. Okay, and for the replication, in case of replication, there is no restriction, and the number of replication will differ from treatment to treatment. Suppose we have taken 5 treatments, suppose A, B, C, D and E, and we generally take 4 replications. That means we will uh, allocate or replicate them, or we will rearrange or repeat that treatment 4 times each. Okay, suppose we are having 5 treatments like nitrogen dose. We are taking 5 different nitrogen dose and we are allocating or repeating them 4 times in the field. Then what we will get, that is a number of plot will be 4 into 5, that is 20. Then if we divide the, if this is the field, we can divide just into 20 number of small plot like this. Okay, 1 2 3 4 6 7 8 9 10 14 15 16 17 18 20. Now let's, in case of randomization, what we do, we divide, we take it as a randomly. Okay, we randomly choose 4 number. Suppose for treatment A, it is the, suppose nitrogen and dose, suppose 40 kg per hectare. We are allocating this treatment A in 4 positions, for replications. So you have to just choose 4 number between 1 to 20. Suppose I have chosen 3 7 13 and 19. We will just put them likewise, like 3 A, 7, 13 and 19. Okay, so we are just putting them in that place in randomly. In similar, in case of like B, we are just taking 4 numbers like 4 or 9, 15 and 17. Okay, so we will just put them in 4, 9, 15 and 17. No such rule about this allocation. Okay, similarly what we can do in case of also we can do in case of um, C, like C, 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 C. In case of D we also can do, in case of D, 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 T, and of course for the E it's the same way we use this allocation. Okay, okay, this is how we can allocate or randomize the completely randomized design. Okay, in this case what we can see that there is the two components of variation. First one, there may be the variation in the treatment. That means if we apply 40 kg nitrogen, and in second one, suppose B, if there is we are applying 50 kg per hectare, there must be present in some variation. So for that reason, there will be present some variation due to treatment, and there may be some experimental error too. Okay, which is common to all, and there is no local control is adapted because the field is homogeneous, so there is no local control is adapted in CRD. Okay, and one thing to remember that the completely randomized design is only used for field experiment, only used for pot cultural experiment or lab experiment, no use in case of field experiment. Okay, let's move to the most important experimental design which we use in very frequently. There is the randomized block design in case of field experiment, that is the RBD. Okay, what is the condition of use? The condition of use is the fertility variation in the field will move from one direction. That means if this is the field, there will be a movement of fertility from this direction to this direction. That means here may be high fertility and here may be low fertility, and this movement of fertility will be just one side, from this side to this side. Okay, if this is the case, we can use the randomized block design. Okay, so what is the layout? We just keep what we do, the field we divide into homogeneous blocks with into equal number of replications. Suppose the replication number is suppose 4, that means we will replicate our treatment in 4 times. We what we will do, we will divide our field, sorry, uh, what we will do, we will divide our field into four homogeneous blocks, 4 homogeneous blocks. Okay, suppose it is block 1, block 2, block 3 and block 4. Okay, and each block we will divide into some small plots equal to the number of treatments. Suppose our equal to the number of treatments, if it is 10, then we will divide the blocks into 10 some small plots. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. In this way, we will then randomize. There is no restriction in case of replication, and the number of replications should be uniform for all the treatments. Here we are supposing the number of replication is 4, and we are allocating the treatments. Number of treatments are the 10, and the replication wise or block wise we are allocating the treatment. That means if we have 10 treatments, uh, 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. We will write them like randomly 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. In this way we will divide our all the blocks, and we will allocate our treatments too. Okay, okay, let's suppose here is the number 5, 6, 7, 8, 9, 10, 2, 3, 4, 1. Okay, this way we will allocate all the treatments in all the blocks and we will have the uh, design. Okay, what are the components of variance is present here? The three. There is a, in the completely randomized only there is a treatment and error. Here we can see there is a block, that means there is a variation or is present in the block wise. That is, if there is it is the far field, we are seeing there is a uh, heterogeneity is present in the field from one direction, moves from one direction, and we are dividing in them into the blocks, homogeneous blocks. That means we can see in this block 1 there is a high fertility, and in this block 3 there is a less fertility, and in this one is medium fertility. That means there is a variation is present into block 1 to block 3, block 1 to block 2, block 3. That means the variation is present between the blocks. Okay, there is a first number, and after that there may be that there is a number of treatment we are using, and there must be present some variation in them. If it present, then the variation will cause due to treatment, and if there must be some accidental error, that may be counted in case of component of variations too. If we see the analysis of variance table for our RBD, we can see what are the number of replication was 4. Then what are the degrees of freedom there will be? There is a n minus 1, that is, here is the r minus 1, that means 3. What are the number of treatment? That was 10, and here that it will be t minus 1, that is the 9. And what will be the error? Sorry, what will be the error? Error will be r minus 1 and t minus 1, that is 3 into 9, that is 27. And if we see what is the total variance will be RT minus 1, that will be something like 39, and we can just add them, 3 plus 9 and 27, we will get 39 too. And if we see the standard error SE, that will use the formula 2VE by N. Okay, V for variance, E for error, and N for number of treatment we use. Then if we see what are the efficiency in case of RBD, efficiency we can use up to 20 treatments. Okay, it is the maximum, or we can use more than that, but there will be not so much efficient. Okay, and RBD is mostly used in case of agricultural field experiment. Then we discuss about the lattice square design. There is the LSD. When we use this design, there is, if there is a present in the fertility variation in two direction. That means the fertility direction will be from this side to this side, and maybe this side to this side too. That means there is a two side of variation is present. Okay, suppose in this side one variation is present, and another side there must be variation is there in this side. Okay, so in that case, what we do, we use the lattice square design for our experimental purpose. Okay, what we do for the layout, we in the layout we just divide our field into two equal number of rows and columns. Okay, the number of rows and columns will be the equal to the number of treatment. Suppose if the number of treatment is equals to, suppose, say 5. Okay, then the number of row and number of column will be as same as the number of treatment. Okay, so what we do, we actually divide them into, sorry, so we divide them into equal number of rows and columns. That means what will be the number of rows and columns? It is the 5 row, and it is a column 1, 2, Okay, this side is the row, and this side is the column. Okay, so there is a 5 row, suppose 1 2 3 4 5, and likewise there is a 5 column, that is 1 2 3 4 and 5. What we do, we do randomization. What we do in case of randomization, we do it is a in using each treatment, we use randomization, using each treatment should occur only one in each row and column. That means, suppose in case of there is a 5 number of treatment, that is A, B, C, D and E. What we do, firstly we allocate them, suppose A, B, C, D and E. Okay, B, C, D and E. C, D, A, D, E, A, B, E, A, B, C, A, B, C, D. Here we can see that each treatment, suppose A is present here. In this row there is only one A, and in this row also there is one A. So, okay, so each treatment should occur only once in each row and each column, and it will form a perfect square shape structure. So we call it as a lattice square design. Okay, so in if we see the number of components of variation, we have seen there is a variation in if we see the field, variation is present from this side to this side and this side to this side. That means from row to row and column to column. That means from row to row, column to column, and there may be present variation in the treatment, and there may be some experimental error too. Okay. Then we will discuss about the split plot design, that is the SPD. And when we take this consideration, when there is a presence of several factors and we are studying several factors simultaneously, we use the split plot design. Okay, what we do, that's the field is divided into main plot, subplot and ultimate plot. Suppose in this case, suppose our first factor, suppose is the irrigation. Suppose our first factor is irrigation. What we do, we divide it into 3 different main plot, that is MP1, MP2 and MP3. That means we are allocating them in the larger area. We are assessing them in larger area. Okay. And next our treatment, suppose it is the nitrogen dose. We what we do, we divide them into subplot. Okay. What we do, we divide them into small subplot. Okay, so what we'll do, it is the SP1, SP2, SP3, SP4. Okay, after that, suppose there is a number 3, that is the seed rate. We are studying the seed rate. for smaller area, we divided them into ultimate plot. We first divide irrigation into main plot, after that nitrogen dose into subplot, and after that we divide the subplot into small ultimate plot. Okay. Okay, so this way we divide our several factors or several treatments into main plot, subplot, and ultimate plot, and we study, and which we will call as split plot design. Okay, you just read this and you will get all the things countable. And in the replication, we don't use any restriction, and number of replication will be uniform for all the treatments. Okay. After that, if we see what is the randomization is the first factor is randomized is the block wise. The second factor is randomized the main plot wise, and third factor is what is the subplot wise. Okay, but here something is different. That is not block wise; it is the main plot wise, and second factor will be the subplot wise, and third factor will be the ultimate plot wise. Okay, and there is the component of variation will be several components, like there may be difference dividing, such as main factor, main plot wise difference may be there, subplot wise difference may be present there, the ultimate plot wise may be difference present there. Okay, that is why the treatment wise also they are experimental, also they are present. And if we see, suppose here in a in a ultimate plot there is a different multi MP1 is there, and it is between SP1, different component is overlapping, so we can say their combination of factors which are creating variation in this case. Okay. Then our design is a lattice design, which we will call as a incomplete design. Okay, incomplete design, and when we use that, there is a sufficiently large number of material is there, like up to 100, up to 200 number of material is present. Then we use this lattice design, and what we do, we just divide our field into some homogeneous plots equal to the number of replication. Equal to the number of replication, we divide our field into homogeneous parts. Then in each part, divide them into plots in equal size in such a way that the number of plots form a square. Okay. So in this case, replication is, there is no restriction, but usually we use 4 number of replication. What we do, we just divide our field into 4 or replication, divide our field in equal equal part, like likewise, and there is a replication 1, replication 2, replication 3, and replication 4, and we allocate our treatment like 1 2 3 4 5 6 7 up to, like here 20, like here suppose 20 19 19 15 16 17 up to 20 number. Here such a we allocate our, suppose there is a number of treatment is 20, we allocate them in 4 replications. Okay, and here we can see the randomization is going in the replication wise, not row or column wise. Okay. The components of variation is present on replication wise, which such that we are uh, randomizing our treatments replication wise, so there may be present in the replication wise. Row and column wise variation is also present. Treatment, there may be variation present, and that also be present in the experimental error. Then our augmented design, it was given by the scientist Federer, and when we use, then when there is a large number of germplasm is there, but the area is limited. When there is a large number of germplasm line is there, and there is also present the, yeah, a huge number of huge area, we use the lattice design, but when there is a limited area we use the augmented design. Okay, what we do, we allocate or replicate the check varieties. Okay, the check varieties we will allocate, but the germplasm line or the treatments actually we don't replicate. Suppose we are using the number of replication will depend on the number of check varieties. Suppose there is a 4 check varieties is there, and we are giving 4 check varieties is there, and we will use minimum 5 replication to them. So what we do, replication on 1, 2, 3, 4 and 5. We will 4 check like A, B, C, D and E. What we do, there is a check and we go for replication on them. A, B, C, D, sorry, 4 check A, B, C, D, A, B, C, D, A, B, C, D, A, B, C, D. But in case of our germplasm line or actually the treatment, suppose it is the about uh, 20, we don't use go for replication. We just allocate them like 6, 10, 5, 3, 4, 2, 1, 7, 9, 13, 15, 17, 13, sorry, 14, 12, 11, like that. Here is the germplasm line. The points to remember that the check is replicated, that is AA is replicated in 5 times. That is how all the checks are getting replicated, but the germplasm lines are not replicated. Uh, then we have discussed the randomization. That is the check. First randomly allocate to the plots in each block, and then a germplasm line that is randomly allocated in the remaining plots in each block. They are not replicated. Okay. If we summarize all the things which we have discussed till now, that about 6 experimental design we have discussed, we see about the conditions of use when we use the CRD, that is when there is a experimental material is limited and the field is homogeneous. When we use the randomized block design, there is a fertility variation of the field in the one direction. When we use lattice square design, there is a fertility variation in the field, there is a move to 2 direction. When we use split plot, when there is a several factor and we will be tested simultaneously. For lattice design, we use there is the number of treatments at large and from say square. Okay, and there is the area also be huge. In case of augmented design, what we see, there is a large number of germplasm, but there is a limited number of area, limited space. Then if we see the local control, the principle of local control is not adopted in case of CRD. There is no local control. In case of there is the error variance can be only be controlled by selecting homogeneous set of experimental design. If we see in all the cases of the experiment, there is a error variance which we can control or reduce by selecting homogeneous set of experimental units. That means what we select the field, it will be homogeneous in terms of their fertility or the experiments we are going to take place. Selection of homogeneous unit is not possible when there is a treatment is large. If the treatment number is large, there is not possible to select those homogeneous units actually. In other designs like RBD, LSD, SPD, lattice, the principle of local control is not adopted by forming homogeneous block. What we can do now, we can divide our plot into homogeneous small blocks as we have discussed earlier, and we can control or we can see what is the main theory, that is the small area. There is a large, if it is the large area, then there is a large variation. If the small area, then will be small variation. Statistical analysis, if we see that the statistical analysis is easy in case of CRD, and but always remains easy even when the results of some units are also missing. Suppose there is some units of some treatment data is missing, then also it is easy. But in case of RBD or LSD, if we see, analysis is easy, but become complicated when missing plot, when there is a some units of data is missing, then we can use the missing plot technique to analyze the data. In case of LSD, there is analysis is also simple but become complicated when there is a several plot yield is missing. In case of SPD, the analysis is difficult than compared to the other design. It is difficult because we are dividing or we are taking into account different number of a huge number of components of variation. If we see the components of variation, in case of CRD, there is a total variation divided into two components. There is a treatment and there is the error. In case of RBD, there is one extra component. There is one is the block. In case of LSD, we have two components. One is the four components. One is the row column, treatment, and error. In case of lattice, there is a five component: replication, row, column, and treatment error is also there. In case of SPD, there are several components present. Like there is a main plot, subplot, ultimate plot, different number of treatments and their combinations too. If we see the most effective one, that is what is the most accurate one. CRD is only used for pot culture experiment. RBD provides the more accurate result than the CRD because it forms homogeneous blocks and separate the randomization in each block. But in case of LSD, we saw the highest number of accuracy because it is fertility variation is controlled in both ways. There is a row and is the column. Here is only controlled by blockwise, and here there is two, that is row and column wise. Okay, so here is the accuracy will be most. It means LSD, in case of LSD, they will be more accurate than RBD. Okay, because in case of LSD, there is a fertility variation is controlled in the two direction, but in case of RBD, there is only one direction. Okay, but RBD is greater than CRD because here we form the homogeneous block, which we will not form in the CRD. Okay, so in case of SPD, there is a provides the accurate precision of different factors because we are using different combinations of factors we are studying simultaneously. Then if we talk about in terms of efficiency, we would see that different designs carries different deficiencies and that will be depending upon the number of treatments. In case of CRD, there is a large number of treatments can be tested, but it will be only lab in pot. Okay, but in case of RBD, the efficiency can be about 20, but in case of LSD, we can see it is up to 12 only. Okay, this is how we can finish our lecture. And suppose we have finished two lectures in this biometric techniques, and we'll going to see more and more. And if you like this video, please subscribe my channel and share with your friends. Thank you.